Alright, here in this video we're gonna end with the second part of mitosis. So how is the cell cycle regulated? So how do cells know when to divide? And just as important, when to stop. When to divide and when to stop. So that decision is really at the center of regulation of the cell cycle, moving through that pie diagram that we'll see in a little bit, and then just getting started at the very, very brief intro to meiosis, going through some more vocab, some more genetics, vocab type stuff. Alright. So let's move on. So the cell cycle is controlled by chemical signals. So moving from one stage to the next is regulated by chemical messengers or chemical signals. So if we remember kind of the chart where we have G1, S, G2, and then mitosis, and the portions outside of mitosis were in interphase. To move from G1 to S to G2 to M, these signaling molecules are required to make sure that everything in the previous stage is completed before moving on to the next, and to make sure that the external and internal environment is conducive, is ready for cells to divide. So first, different types of cells divide at different rates. So the lining of your stomach, those cells get regenerated very, very quickly because of the stomach acid that constantly wears away at them, so they have a high rate of mitosis. On the other end, muscle cells, like we have here, these are cardiac muscle cells, and nerve cells, or neurons, never divide. So once they mature and are specialized, they never really go through mitosis after that. So molecular signals tell the cell when to divide and when not to. So for example, if we have a whole bunch of these cells all bunched up and there's really no space for them, it'd be important for those signals to tell these chemical messengers to tell these cells there isn't enough room to divide and make more cells. We need to stop. We just need to stay for a while, you know? Or for example, in the case with your skin, if there's not a break in your skin and you're not really growing too much, they don't need to divide as fast. Those chemical signals or messenger molecules are responsible for communicating with all the skin cells to tell them that. Experiments have been performed that verify that in fact it's chemical signals or messengers that are responsible for regulating the cell cycle. And this is a really cool experiment. So what scientists did is took a cell that was in S phase, so duplicating its DNA, and a second cell that was in G1 phase, or just going about its normal cellular processes, only one copy of the DNA, and they fused them together, so their plasma membranes became one. They fused them together, and what they saw was immediately this cell that previously was in the G1 phase immediately jumped to the S phase and duplicated its DNA. So immediately on contact. Same time, they took another cell that was in mitosis and fused it with a cell that was in G1 phase. And when they were fused, the G1 nucleus immediately began mitosis. Spindle fibers were forming, even though the chromosomes didn't duplicate. So the chromosomes didn't duplicate, we didn't duplicate our DNA, but immediately it started mitosis, cell division. That supported this hypothesis that there are chemical signals present in this cell and this nucleus that then, when they're fused, migrated over and told this cell something specific to enter an S phase, or in here, told this G1 cell, we're going to start going through mitosis right now. And that is the case. We have different chemicals for different stages in the cell cycle to start or stop different processes. So what's important is the cell cycle has checkpoints, or little stop signs. So we're going around the cell, and we have to pass these checkpoints, be like, yep, everything checks out to move on to the next. Yep, checks out, everything moves on to the next. These checkpoints are placed where the cell cycle is stopped to make sure everything's in order before the cell cycle continues. So generally, when the cell hits one of those checkpoints, it stops what it's doing, its progression through the cell cycle, and doesn't continue until it receives a specific message, a specific chemical signal to continue. So for example, in the G1 phase, we reach the G1 checkpoint. And the cell makes sure that its size is appropriate, it has enough nutrients, enough growth factors, and that the DNA isn't damaged. And if all of these things can be checked off, a chemical signal lets the cell know that it can proceed to synthesize DNA into the G2 phase. And then another checkpoint happens in the G2 phase, making sure, yep, we have enough room to duplicate, our DNA is replicated, no errors, and then it goes to mitosis. And in mitosis, there's then a third checkpoint. When the spindle fibers and metaphase are all connecting to the kinetochore of the chromosomes, they check to make sure that all the chromosomes are attached to spindle fibers before anaphase starts. And once the signaling molecule is present, it can continue on. If at any of these points it's a no-go, we don't get the go-ahead signal, the cell exits the cell cycle into something called G sub 0 or GO phase, where it's just kind of out dormant, it's not going through this continual process of mitosis. So these are the major things that the cell looks for at each of these points. Alright? So, for example, if we check off, yep, that all chromosomes are attached to the spindle fibers, then an enzyme called separase can cleave the cohesin protein bonds in between the sister chromatids of the chromosomes, and they can separate and go through anaphase. So these chemical messengers come in a lot of different forms, and some of the important ones that are discovered in 2001-2002 were cyclins and cyclin-dependent kinases. These are the chemical regulators of the cell cycle. So cyclins are a major group of signaling proteins that regulate the different checkpoints of the cell cycle. Their presence, so having the presence of a cyclin, activates a cyclin-dependent kinase, or CDK. Remember, kinases phosphorylate different molecules. So, normally, say we have a cyclin-dependent kinase that's going to initiate S phase. It's in the G1 gap phase. And normally it's not active. It can't phosphorylate these target molecules to start the replication of our DNA. Well, at the right time, when all those factors are checked off, a cyclin binds with the cyclin-dependent kinase to form a cyclin CDK complex. Then this cyclin-dependent kinase becomes active. It can phosphorylate these different targets, and the S phase can begin. DNA replication can start. They're called cyclins because their various levels inside the cell tend to move cyclically in response to the cell cycle. They vary based on the stage of the cell cycle. So this G1 cyclin is at its highest towards the end of the G1 phase, and then it goes and is decreasing, and then it increases as the G1 phase approaches again. So that's why they're called cyclins. Their concentration is in a cycle like this. They're very important for regulating when things happen and for moving from one gap phase to the next. G1 checkpoint to G2 checkpoint to M checkpoint. Alright? And here is a diagram of how that looks. So our G1 cyclin D, as we progress through the G1 phase, rises, and then all of a sudden cyclin E increases in abundance to usher in S phase. And as S phase progresses, its concentration decreases, and cyclin A's concentration decreases and enters in G2 phase. And as G2 goes on, its concentration decreases, and cyclin B increases to start triggering mitosis. So you can see that they cycle, their concentrations cycle depending on the stage of mitosis. So there's other factors that influence cell cycle regulation besides cyclins and cyclin-dependent kinases. Other factors, both internal and external, influence the cell cycle. Internal, like the M phase checkpoint in the spindle fibers joining the kinetochore. So the spindle fibers have to be joined to every kinetochore for it to move forward. That's an example of internal regulation. And external regulation, the presence of nutrients in the growth media enable the proliferation of cells. So if you take human blood cells or things and you put them on a growth media, like in a petri dish, and there is not any growth factor, no nutrients, or not the right nutrients, we'll have no proliferation, because those nutrients are a external cell cycle regulator. If those nutrients aren't present, it tells the cell not to duplicate, not to reproduce. Also, other growth factors, so chemicals that cells can give off to each other that they need to stimulate growth, are some other external influencers on the cell cycle. We also have density-dependent inhibition and anchorage-dependent inhibition. So most cells in living organisms must be anchored or attached to something for cell division to start. So they can't just be floating in water and start, even if that water has all their nutrients, can't be just floating out there to start dividing. But they either need to be attached to like the side of a beaker or part of a nutrient plate, or to the extracellular matrix of other cells in your body. So it is anchorage dependent. It must be anchored to something. In addition, once they start dividing, they'll divide and divide and divide until they're surrounded by other tissue. There'll be a flat layer. And once they sense that there's cells all around them, they won't divide any further. That's what's called density-dependent inhibition. So the cell cycle is cut off if they sense there's other cells around it. That has to do with a lot of the membrane-bound signaling proteins like we talked about to signal there's other cells out there. In fact, if you do a test like this and remove some of the cells close to that hole will start dividing again to fill it in. The last example is platelet-derived growth factor. So if you get a wound and some platelets in your bloodstream are traumatized, they will start to signal the division of more platelets to patch up that cut, to form that clot, and then when it's formed, platelet-derived growth factor won't be released from the platelets anymore, and they'll stop dividing. So you might have noticed that one of these big implications for this in the cell cycle is cancer. Cancer cells are an example of when the cell cycle regulation has gone very wrong. When checkpoint mechanisms are overridden, cancer cells do not display density or anchorage dependent. So when mutations happen in DNA for cells, it might totally override those checkpoint mechanisms, and the cell will keep dividing, no matter what's outside of the cell. So cancer cells aren't density dependent. Even if there's cells close to it, they'll keep dividing and create this huge mass, this tumor. And in malignant tumors, they're not anchorage dependent either. They can break off into the bloodstream, the lymphatic system, and keep dividing and grow and metastasize. So cancers do not stop dividing when growth factors are depleted. Many, in fact, manufacture their own cyclins independent all the time. Doesn't matter what stage of the cell cycle it is, so the cell cycles keep going and going. What happens because of this is most cells stop dividing or die through apoptosis after 20 to 50 different divisions. That's not the case for tumor cells or cancer cells. They'll keep going and keep going, almost becoming immortal. So that was kind of the end of mitosis. So that is the end of the unit. So everything up to this point, it can be considered on the exam. Anything after this, or the next slide, not on the exam. With that, I want to just take a little bit to introduce a little bit of meiosis that we'll be looking through next. Just a second. To get us started. So, in meiosis, what meiosis is the creation of gametes. So those are sex cells, those are sperm, and those are eggs. So in meiosis, we're creating sex cells, sperm and egg, and they both have half the total genetic info. So in humans, sperm and egg cells only have 23 chromosomes. And what the sperm and egg cells do is carry genes. So these are the smallest unit of inherited information that's passed along from parent to offspring in sexually reproducing organisms. So each gene is located at a different position or a locus along a chromosome. So if this is chromosome 1, this is our other copy of chromosome 1, we might have a specific position right here, a locus, where the gene for, let's think, where the gene for sickle cell disease is located. That is a specific gene, a specific piece of inheritable information that's found at one spot on a chromosome, and that's called a locus. Parental genes are passed to offspring via these gametes, these sperm and egg cells, in sexual reproducing organisms. In asexually reproducing organisms, prokaryotes that reproduce through binary fission, offspring are genetically identical because they don't create gametes. No gametes. So each offspring is genetically identical to the parent. Each prokaryote's offspring is a clone of the parent. Alright, so in the next class or two, we'll look at meiosis, how those sperm and egg cells are created to only have half of the genetic information. Alright, that's it for today.